All right, you're live.
Okay, so for number one, it asks about the banding pattern on the gel. Um, there's four lanes for GTP, ATP, TTP, and CTP. Um, each of the lanes have three bands, except for TTP, it has four bands. Just try to show it right now. But that's how I have them being spread out from the largest molecules on the top and then the shortest ones down at the bottom. With the positive end at the bottom and the negative at the top. Um, for part B, DNA polymerase will denature because of the high heat, while Taq polymerase is supposed to be resistant to that heat, and so the reaction won't occur because synthesis can't occur without that enzyme. For C, DNA helicase isn't necessary for the reaction. The purpose of separating the molecules is the heating step, and so you don't need DNA helicase, but it will denature, but synthesis will still occur there. For D, so I wasn't sure if there were other nucleotides present, and if there were, then you would end up just having an entire molecule synthesized, and then there's only one band on the gel. But if not, then without the DD NTPs in the other three tubes, new strands can't be synthesized at all because there's nothing to add to the base to the molecule to be synthesized, and so synthesis won't occur in that case.
Okay, so for two, it's just fill in the blanks. So A is during a typical PCR cycle, the GAPA synthesis step is done at 72 degrees Celsius, while the priming step is done at 55 degrees. B is AMPR is a common selectable marker that is found in many plasmid vectors, while restriction enzymes recognize and cleave DNA molecules at palindromic sites. C is during agarose gel electrophoresis. DNA is placed in electrical field, so DNA migrates towards the positive electrode during electrophoresis. I have a hard time saying that. And then for D, DD, ATP lacks the 2 OH group and the 3 OH group. And then E, transformation is a term used to describe the process where bacteria take up DNA molecules from the environment. And E, during blue-white screening, IPTG is commonly used to induce the expression of the lac operon, while EPSGAL is cleaved by B-galactosidase to turn E. coli cells blue. So 3 is talking about using PCR to amplify the region of the double-stranded DNA fragment from E. coli. And then it just tells you the sequence is copied, that's copied and located between the sequences below. And then it asks us to write out a single-strand DNA sequence that would be used for the 2 primers in the PCR experiment. So for primer 1, we had 3' CTC, GG, AA, GTG, AG, TC, AT, and then 5'. And then primer 2, we had 5', TCG, GHG, TTA, ACG, PG, AC, and then 3'. And then for like the mathematics of it all, I'll just show my math, I guess. That's my math. But we got, for the primer 1, we got like 59 degrees Celsius, and primer 2, we got 62.4 degrees Celsius. And then for C, it asks us what the sequences of the newly synthesized strands of DD-CTP is. And then, so the first one we got was 5', TTT, AA, TC, and then 3'. And then for the second one, we got 5', GGG, A, TT, AG, and then 3 prime. And then the next question is, when you learn about DNA replication in E. coli, you'll learn about DNA and DAM, and then why are these 2 proteins not included in a PCR experiment? And we just said that it's because, hold up. Because they're generally used in denaturation, and that's probably why they're not included. Yeah, and it's through like high temperatures and stuff.
So for question number 4, again, I'll just try to show my picture for this. I don't know how else to do it, but this is what I have for how the bands will separate during electrophoresis. So again, with the positive end at the bottom and the negative end at the top, and so the smallest molecules will travel the furthest to the positive end. For the restriction map, showing the locations of the restriction enzyme sites, I'll just show that one again, but there's the band, and then there's, which one's next? And then the EcoR1. So, no, I should explain that. But, and then for part C, there's going to be a 500 base pair molecule is the largest. Then there's a 150 base pair. Then there's a 100, and then there's a 50 base pair if all of the molecules are present.
Right, so with questions 5 and 6, question 5 is asking to detail the steps that you would use to generate cDNA from Saccharomyces cerevisiae, a mature mRNA. Clone the cDNA version of YFG into a plasmid vector and use the blue-white screen to identify the plasmids that contain the YFG cDNA. So in order to get the cDNA, you'd use the viral enzyme reverse transcriptase. Next, using the restriction endonuclease for both YFG and the plasmid vector, the genetic material could recombine. Using heat shock, the receptor cells could be induced for transformation. The host DNA would be a mutant lacking the LAC-Z gene. If the plasmid took up the recombinant DNA, then LAC-Z gene would not be present on the plasmid. In this case, no beta-galactosidase could react with the XGAL to produce the blue pigmentation. And so that's kind of a summary of the steps that you would use to get what you want in question 5. Question 6 is asking about CRISPR and Cas9. It's a modern technique. It's used to basically edit someone's DNA or something's DNA. It occurs naturally in bacteria in order to prevent viruses from attacking the bacteria. It's basically taking up viral DNA in segments in order to protect against that virus. And so in this way, the DNA is replicating that DNA in order to use it as a defense mechanism. Cas9 can cut the foreign DNA when attacked. Let's see. It's used by scientists because CRISPR/Cas9 can cut DNA at specific target sequences. This holds hope for preventing and treating diseases common to humans like cancer and other genetically derived diseases. Thank you very much.